So 16.15, I'll start by reading the question. Milk products such as cheeses and yogurts are dependent on the conversion by various anaerobic bacteria, including several lactobacillus species, of lactose to glucose and galactose, ultimately producing lactic acid. Okay, so let's start by mapping out some of this here. Looks like it's going to be a long question. Okay, lactobacillus. So lactobacillus bacterium is going to take lactose for us. Kind of sounds like it's going to be a lac-operon question. It's going to break down lactose into glucose and galactose. Okay, well that's a good thing, I guess, if it's going to make cheese and yogurt. These conversions are dependent on both permease and beta-galactosidase as part of the lac-operon. Okay, so lac-operon diagramming here. We have lac-I separated from the promoter, operator, lac-Z, which is beta-galactosidase, lac-Y, which is the permease, and lac-A, which is, we don't know yet, promoter, operator, repressor binding site, and the repressor. Okay, so after selection for rapid fermentation for the production of yogurt, one lactobacillus subspecies lost its ability to regulate lac-operon expression. So it seems like what they did was they took some bacteria and they selected for mutation or something, some subpopulation that eventually became a subspecies that lost the ability to regulate. Oh, they were selecting for rapid fermentation. So, because they need rapid fermentation to make yogurt, and during the selection process, they isolated one subspecies. I don't know why they call it a subspecies and not a, different strain, a mutant strain. They call it a subspecies, but okay. A lactobacillus, let's call it a mutant. I don't like subspecies here. Lactobacillus mutant that lost ability to regulate lac-operon expression. Okay, cool. That seems like that'd be a pretty valuable mutant strain there, especially if it led to rapid fermentation of yogurt. Okay, you can make a lot more yogurt in a smaller amount of time, or a lot more yogurt in the same amount of time, or the same amount of yogurt in a quicker time. But you get the point. So would you consider it likely that in this subspecies, this mutant, the lac-operon is on or off? So is it always on or always off? Okay, so we know that it lost the ability to regulate the lac-operon. So we need to figure out if the operon now is always on or always off. And this is kind of too simple, right? I think this is way too simple, because unless there's something I don't understand, which is completely possible. But if the purpose is to break down lactose into glucose and galactose, and the mutant is working better than the wild type strain. Well, the wild type strain can sometimes turn, it's usually off, and usually the lac operon needs to be induced. Turn this on. So I'm guessing that it's now always on. So it doesn't matter. It doesn't matter if lactose is there. This operon is always going to be on in the mutant. Beta-galactosidase, LAC-Z, which is needed to turn lactose into glucose and galactose, is always going to be expressed. And yeah, so it's going to make this bacteria much better at fermenting, and it's going to make it ferment much more rapidly. So we're going to consider that in this mutant, the lac operon is always on. And the next question is, what genetic events would likely contribute to the loss of regulation as described above? And if you did the other problems, you probably already know what possible mutations are involved. Either we have mutated the repressor binding site, so this is LAC-OC. Now it's constitutive. The RNA polymerase can always bind to the promoter and always transcribe these genes because the repressor can't bind. Or the mutation could be that we've mutated the binding site for the operator on the LAC-I repressor, so it can no longer bind the operator. And as a result, the RNA polymerase will be free to always transcribe the operon. Those are some possibilities. And we could also consider some others, considering the cap binding protein, right? I guess we could, for the cap binding protein, which would be on this side of the promoter right here, and I can put the cap binding site in there. So this is the cap binding site, and cap binding is when it binds, it enhances the ability of RNA polymerase to bind the promoter. And perhaps we could have a mutant cap protein that is always bound to the cap binding site, so it doesn't need cyclic AMP anymore. Okay, that would be a possibility too. Or maybe we can even have a mutation in adenyl cyclase. So adenyl cyclase now is always converting ATP to cyclic AMP. And if cyclic AMP is high, it'll always be bound to the cap activator protein. And then the cap activator protein will always be bound to the cap binding site. So there are a number of mutations that could lead to this rapid fermenting mutant of Lactobacillus bacteria. I think probably the better ones, the ones that are easiest to understand, are the mutations involving LAC-O and the LAC-I repressor.